Hello. In this video, we will be depicting the plague disease through the immunity model. The plague disease boomed during the Middle Ages and killed millions of people primarily in Europe. Although antibiotics and protocols have dealt with the virus in most places, there are still a few places affected by the virus. The plague is caused by bacteria, Yersinia pestis. The bacteria Yersinia pestis is a gram-negative bacillus-shaped bacteria that is suited to multiply in the lymph nodes of humans. People can get infected by the bacteria through inhaling air droplets into the lungs, flea bites, or touching the tissues of infected animals. Due to unhygienic practices of the virus, close quarters, and a high population of rats, the virus spreads rapidly throughout the body. One can diagnose themselves through a lab test using their pus. There are 3 different levels of the plague: the bubonic plague, pneumonic plague, and septicemic plague. The first level is the bubonic plague, where the bacteria is localized to only one region of the body. The septicemic plague is when the bacteria spreads to multiple regions or systems of the body throughout the bloodstream. The pneumonic plague is whenever the bacteria infects the lungs. Now let's get into the model. Here we see the rodent, which in our case is the rat, contracting the Yersinia pestis bacteria through ingesting infected food. A flea then sucks the blood containing the bacteria. The bacteria then multiplies in the flea's gut, making it hungry. The human tries to attack the rodent, but while it is distracted, the flea travels to the human host and feeds on the human. To feed on the human host, the flea makes a wound on the skin of the host, which allows the flea's infected blood to also travel into the human and go into the human's bloodstream. The bacteria enter through the cut made from the flea through the flea's bite and produces proteins such as the Yersinia outer proteins, or YOPS, the F1 antigen, and V antigens. The YOPS inhibits different leukocytes by being injected by the Yersinia pestis into these cells. Our model depicts the F1 antigen and the V antigen by displaying themselves as spikes on our bacteria. The YOPS protein is depicted by being injected into various leukocytes. These proteins inhibit the innate responses by stopping the release of histamines from the basophils, preventing the bacteria from being engulfed by phagocytosis by neutrophils or macrophages, hindering the neutrophils' chemotaxis towards the Yersinia pestis, and interfering with cytokine production. The bacteria lead to an overall reduction in natural killer cells. They also result in a reduction in movement and maturation of the dendritic cells. The primary adaptive immune response is affected by these proteins. Since the proteins reduce the maturation of dendritic cells and production of cytokines, they interfere with the activation of helper T cells. The helper T cells cannot activate and present antigens to the B cells, meaning that the B cells cannot become notified of the foreign bacteria and differentiate into plasma cells to fight against the bacteria. By inhibiting the activation of B cells and helper T cells, Yersinia pestis interferes with the secondary adaptive immune system. Since the B cells are not notified of the bacteria's antigen, they are unable to clone themselves to create memory B cells to create the necessary antibodies to fend off the bacteria. So when the person is reinfected with the disease, even if there were memory B cells which could fight against the bacteria, without functioning helper T cells, they are rendered useless. With no memory B cells being produced to be able to activate or defend against future infections, the human body will have the same response that they had the first time, making this disease very deadly and difficult to contain. There are many symptoms that occur from the plague, including fever, chills, headache, muscle aches, and fatigue as the body tries to fight off the bacteria. But the most distinct symptoms shown in humans is buboes. Buboes are sores that occur because of lymph nodes swelling up. These swollen lymph nodes can spread through the body, causing septicemic plague. The lymph nodes lead to tiny blood clots around the body that start to kill tissue around the body because of the lack of circulation that it brings. This is also known as necrosis. If left untreated, the host eventually dies, as seen in our model. This disease was increasingly prevalent in the Middle Ages because of the lack of knowledge that was available at the time. In modern times, it is easily preventable due to the magic of antibiotics. Antibiotics essentially stop the DNA replication and transcription of the bacteria Yersinia pestis, thereby inhibiting the growth of the bacteria. Some antibiotics that are helpful are streptomycin, gentamicin, and doxycycline. In addition to antibiotics, there have been many control measures, such as the rodent control, flea prevention, and practicing good hygiene to prevent the spread of this terrible disease that struck disaster in the Middle Ages.